Thank you. Ingrid Swanson Pultz is a senior fellow at the Institute for Protein Design in the School of Medicine, working on the development of oral treatments for celiac disease. She's a founder of PVP Biologics, a company formed to bring this remedy type of therapy to market. She has her undergraduate degree from Wellesley College and completed graduate studies in microbiology here at the University of Washington, where she founded the UW International Genetic Engineering Machine, iGEM team, which I hope she can describe. Her topic today, an area of really intense focus in the popular literature, is on the topic of gluten and celiac disease. Ingrid, thank you.
Thank you. Thank you. So my name is Ingrid Swanson Pultz, and I'm a researcher at the Institute for Protein Design here at the University of Washington. The Institute for Protein Design is an institute that uses computational protein design tools, in other words, software, to make new proteins that don't exist in nature for the purpose of solving global problems. And this can include diseases and also all sorts of other problems, like energy generation or degradation of pollutants. And my group in particular engineers new enzymes for the treatment of celiac disease. And so what I'm going to tell you about today is celiac disease and its trigger, which is gluten. Gluten is a protein that's found in any food containing wheat, rye, or barley, which includes all of the foods that are shown here, as well as pizza and beer, and other sources that you might not know have gluten in them, such as soy sauce or licorice. And before I start to tell you about how we've developed a treatment for celiac disease, we're going to do an audience participation game called Gluten Facts, True or False, in which I make a statement, and then we tell by show of hands how many people think that statement is true or false. So the first statement is that the prevalence of celiac disease is increasing. How many people think that this is true based on what they've heard? How many people think that this is false? Okay, so about 60-40, interesting. So this is true. What's shown is a graph demonstrating the number of new diagnoses of celiac disease over time for a 20-year period of time. And you can see that the number of new diagnoses are, in fact, increasing. But is this due to an increased awareness of the disease or an actual increased incidence? So is it that more people are just aware that this disease exists, physicians and patients, or that the actual number of people getting this disease is larger? How many people think increased awareness? How many people think increased incidence? The answer is actually both. So while it's definitely true that awareness has been increasing for celiac disease used to be thought of as a disease that children get, and that it's specifically limited to GI symptoms. But now it's understood that this is a disease that can be diagnosed at any age, and that the symptoms might not even necessarily present as any GI symptoms. There are all sorts of symptoms associated with this disease, which I'll go into in a minute. But it's also true that the incidence of this disease is increasing. And this is a study that was done at a US Air Force base, which banked about 10,000 blood samples in the late 40s and early 50s. And more recently, a study was done to thaw these blood samples and to look for the presence of antibodies that are specific for celiac disease. And this study found that 0.2% of those original 10,000 people had the disease. And that's compared to 0.8% that was found in 10,000 matched individuals today. And in fact, it's known, other groups have also identified this is the prevalence of celiac disease. So celiac is increasing in its incidence. The reason why celiac disease is more prevalent is because of genetically modified wheat. Now I'm going to actually add a third option to this, true, false, and we just don't know enough information to be sure. So how many people think this is true? How many people think this is false? How many people think we don't have enough information to be sure? That was actually a red herring, because the answer is that this is false. And the reason for this is simple, because there's been no genetically modified wheat that has been commercially available. However, the genome of wheat has been modified by people through hybridization techniques or standard farming practices over the course of the last couple of centuries. So the wheat that we eat today is not the same wheat that was eaten 200 years ago. And whether this is, in fact, linked, related to celiac disease, nobody really knows. Gluten can cause symptoms in people with celiac disease. Without celiac disease. Gluten can cause people with symptoms that do not have celiac disease. How many people think this is true? How many people think this is false? This is actually a complicated answer. What we're talking about is non-celiac gluten sensitivity, or NCGS. And unlike celiac disease, which is a very well-defined, well-characterized disease that has objective ways to diagnose this disease, non-celiac gluten sensitivity is a much more ambiguous disease. So in this study, which was done in 2011 in Australia, patients with self-diagnosed non-celiac gluten sensitivity either ate 2 slices of bread and a muffin that contains gluten or that didn't contain gluten every day for 6 weeks. And symptoms were monitored over this period of time. And what they found was that even patients that ate the placebo gluten food had an increase in symptoms. So there was clearly a placebo effect. However, patients that ate the gluten-containing food had more symptoms than patients that had placebo-containing food, which indicated that something was in fact going on with gluten. However, the same group did another study 2 years later where patients ate 2 slices of bread and a muffin that had either a high level of gluten, a low level of gluten, or no gluten. And it was found that there was no difference between these groups in terms of their symptoms. And in fact, the group that received the high gluten had a few less symptoms than the other groups. So the authors hypothesized a number of reasons for this. There are molecules that tend to be highly enriched in foods that contain gluten, and perhaps these other molecules could be the culprits. And these include FODMAPs and ATIs, which I'm not really going to go into in detail. FODMAPs, though, are sugars that tend to cause people to become bloated and uncomfortable. It could be gluten, and this one study does not necessarily disprove that there's any effect of gluten. It could be the gluten, we just haven't identified it yet. Or there could be some other factor. So that's kind of a long-winded answer to say that this is really unclear what exactly is the basis for non-celiac gluten sensitivity and how many people this disorder affects. Next gluten fact, gluten-free food has no gluten. How many people think this is true? Wow, almost nobody. So I assume the rest of you think that this is false. The answer is that it's not necessarily true. So the FDA says that if you are to label your food as gluten-free, that means that the food has less than 20 parts per million gluten. And that 20 parts per million is 20 milligrams per kilogram of food. So that is not very much gluten. And the reasoning behind this is because if you have celiac disease and you eat food at that 20 parts per million gluten level, you're not predicted to get symptoms. Now granted, there are some people with celiac who could still get symptoms with that small amount of gluten. Also, when you go out to a restaurant, if a food is labeled gluten-free, that doesn't mean that the food necessarily meets this strict threshold. For example, we saw an ad at Pizza Hut that said, now serving gluten-free pizza, not safe for celiacs because it's prepared in the same place as gluten-containing pizza. So that would not reach this level. Next fact, everyone would benefit from a gluten-free diet. How many people think this is true? Okay, how many people think this is false? Most physicians and nutritionists would say that this is false. There's no data that indicates that if you don't have celiac disease, and probably if you don't suffer from non-celiac gluten sensitivity, that you will have no benefit if you eat a gluten-free diet. Look, not eating gluten just makes you healthier, okay? True or false? How many people think this is true? How many people think false? This is true and false. And the reason why is because historically, eating a gluten-free diet meant that you ate a lot of legumes, plants, fruits and vegetables, lean meats. You didn't eat baked goods, and this made for a healthy diet. However, nowadays, gluten-free food, gluten's really an essential component of a lot of baking processes. So a lot of companies will try to substitute the gluten by adding extra sugar or extra fat. And this means that the gluten-free food can actually be less healthy than gluten-containing foods. So most nutritionists would say to just eat a balanced diet. You don't necessarily need to cut out all gluten from your food. unless you have celiac disease. So this is really a debilitating illness. And people who have celiac disease, the ingestion of even small amounts of gluten can result in classical GI symptoms of abdominal pain and diarrhea. And actually, before I get started here, how many people in the room either have celiac disease or know somebody with celiac disease? Okay, so about half of you. This disease, as mentioned earlier, affects almost 1% of the population, so it's a fairly common disease. Celiac disease can also present itself in malnutrition-related disorders because people with celiac disease have inflammation that's caused by the ingestion of gluten. And this inflammation in the intestines, as we've learned quite a bit tonight, damages the intestines, which leads to an inability to absorb nutrients. Celiac disease can also have neurological and dermatological symptoms. And finally, the disease can result in a small percentage of people with the disease getting non-Hodgkin's lymphoma, which is due to chronic inflammation resulting from continuous gluten exposure. And this is a cross-section of intestines. So the intestine on the left is a cross-section from someone with healthy intestines. And you can see these nice, long, finger-like projections that are involved in absorbing nutrients. And the intestine on the right is the intestine of someone with celiac disease who's on a gluten-containing diet. And you can see that there's a large amount of inflammation here. And in fact, these finger-like projections have been completely destroyed, which means that there's not a lot of nutrient absorption going on in this intestine. Luckily for people with celiac disease, going on a gluten-free diet can result in healing of the intestines, although the intestines will rarely get back up to the level of what is considered to be healthy for a normal, healthy individual. And the gluten-free diet is actually very difficult to achieve. So this diet must be kept very strict. In fact, clinical studies have shown that the threshold of gluten for triggering the disease is between 10 and 100 milligrams. And to put this into perspective, the amount of gluten in a slice of bread is between 3 and 4 grams of gluten. And so it's actually very difficult to achieve this because gluten is everywhere in modern food production. In fact, 60 to 70% of celiac patients who are on a gluten-free diet still get symptoms that are due to gluten ingestion. In addition, the gluten-free diet is burdensome, expensive, and life-restricting. And so celiac patients and their doctors have indicated that they would place great value in a therapeutic that could prevent the symptoms and intestinal damage upon gluten ingestion. And this is where the work of my group comes in. So how does gluten cause celiac disease? Gluten is a protein that's a conglomeration. It's actually a conglomeration of 2 different proteins. And these 2 proteins are called gliadin and glutenin. And it's the gliadin proteins that are responsible for celiac disease. And the gliadins, in turn, are made up of 3 different gliadin families, the alpha, gamma, and omega gliadins. What's shown here is the amino acid sequence of alpha gliadin. Amino acids, so recall from your biochemistry classes that amino acids, there are 20 different amino acids, and they can be designated each by a 1-letter code. So this is a long string of amino acids. This is the amino acid sequence of alpha gliadin. This red highlighted region here is the region that triggers celiac disease. And in fact, there's also a region in gamma gliadin and in omega gliadin. So you'll notice that this region has a lot of P letters here. The P stands for proline. Proline is an amino acid that there's nothing inherently wrong with proline, but proline amino acids are just very difficult for human digestive enzymes to break down. And so as a result, gliadin, ingested gliadin goes into the stomach, through the stomach, and into the intestines only partially digested. And then in most normal, healthy people, these incompletely digested gliadin fragments just go right through with no problem. But people with celiac disease mount an inflammatory immune response against these incompletely digested fragments of gluten. And I should mention that, that experts in celiac disease believe that the best way to treat this disease would be the administration of enzymes that would break down these difficult to digest fragments in the stomach before they can get to the intestine and cause the disease, similar to a lactate pill that breaks down lactose. And so when we sat down and thought about making an enzyme for the treatment of celiac disease, we recognized that there are really 5 essential characteristics that this enzyme would need to have. And the first was an ability to function in the stomach. And this is really important because you need to be able to break down gluten in the stomach before it gets to the intestines, which is where it triggers the disease. And the second was the ability to target all of the toxic regions in wheat, barley, and rye. The third is an ability to reduce the amount of gluten present to below that threshold that triggers the disease. The fourth is that it needs to be very cheap and easy to produce. And the fifth characteristic is that it really should be specific for gluten and not act upon other proteins that are present during a meal. Only about 20% of the protein that we eat in a Western diet is gluten. We eat a lot of other protein when we eat. And a lot of this is, for example, dairy or meat protein. And so if you have an enzyme that breaks down everything that you eat, if you eat a lot of other protein, the enzyme will be distracted by all of this other protein and will not focus on the gluten. And so the enzyme that we've developed in my lab is called KUMAMAX. And this is an enzyme that functions to break down these toxic gluten fragments in gastric conditions. And the resulting fragments are no longer capable of stimulating the immune response. And we made this new enzyme. So the reason why a new enzyme is necessary, and you can't just go and pull an enzyme off the shelf and use it to treat celiac disease, is because it's difficult to break down gluten and it's difficult to work in the stomach. So there aren't a whole lot of enzymes that do either of these things, let alone an enzyme that has both of these characteristics in the same enzyme. And this is actually why it made such a great project for protein engineering. And actually, as an aside, I was asked to talk a little bit about the International Genetically Engineered Machine Team. This actually began as an undergraduate student project in 2011, and the students generated a prototype enzyme in the course of 3 months using design software. And what we did was we started with an enzyme called KUMAMAX. So we started with an enzyme that had some of the properties that we were looking for, namely an ability to function in stomach conditions, and then engineered in the properties that the enzyme was lacking. And so this enzyme, KUMAMAX, is produced by a bacterium called Alicyclobacillus sendiensis. And this is a bacterium that grows in acidic Japanese soils and at high temperatures, so conditions similar to your stomach. And this enzyme is well known in the literature. It's very easy to make, but it has no activity on gluten. And that's what the specificity for PR means. It doesn't work on gluten. And so what we did was we engineered this enzyme to break down gluten. And we did this. One of the nice things about working with an enzyme that's very, very well studied is that there's what's called a crystal structure available. So it's 8:45, and I know that probably not a whole lot of you are very interested in the software that we use to design this enzyme. So I'll just go over it really briefly. The green here represents the enzyme, and the purple here represents this tiny fragment of gluten that we've modeled using the software into the business end of the enzyme. And then we made theoretical mutations in the software in and around the active site that are predicted to increase the ability of this enzyme to break down gluten, to rip it apart. And then once we identify some mutations in this enzyme that are promising, we then produce the enzymes in the lab and test them on toxic gluten fragments. We then combine those mutations that are promising, and this is an iterative round of design, building, and testing different enzymes. And so this amino acid sequence right here is that red highlighted amino acid sequence from alpha-gliadin. This is the amino acid sequence from alpha-gliadin that triggers celiac disease. And in particular, there are 5 regions in this amino acid sequence that are underlined that are responsible for triggering the disease. And while your digestive enzymes cannot break down this entire fragment, KUMAMAX is capable of targeting this fragment for destruction at 3 different locations. And these 3 locations destroy all of the problematic toxic fragments of this peptide. And so the breakdown products are not immunogenic. And it does this in also the immunogenic regions in wheat and barley, or barley and rye. This is an experiment in which Clancy Wolf, who's a highly talented research scientist in my lab, went out and bought a loaf of Oro-wheat whole wheat bread, brought it back to the lab, and took 2 slices of bread and mashed it up in what we call artificial saliva, which is an electrolyte solution. And he then acidified the mixture by adding hydrochloric acid. And we then incubated the mixture at body temperature in the presence of pepsin, which is the major digestive enzyme in the stomach, and KUMAMAX. We used a KUMAMAX that we call KUMA30, which is shown here, but it's KUMAMAX. We let digestion happen for 30 minutes, and then we quantified the amount of toxic gluten that was remaining. In this study, we compared the results against bread that has been incubated with enzymes called EPB2 and SC-PEP, so that's that black line on the top. That's the amount of gluten that remains if you treat the bread mixture with these two enzymes. These enzymes are significant because these enzymes are currently in clinical trials to treat this disease. However, these enzymes don't work very well in the stomach, as I mentioned earlier. It's an issue. And so what we find is that at the highest enzyme concentration that we use, enzyme concentration is listed on the x-axis, we see about 85% degradation with these two enzymes. And I know that doesn't look like 85% degradation, but keep in mind the y-axis is on a log scale. At the same enzyme concentration, we see over 99.9% degradation by the KUMAMAX enzyme. And even at lower enzyme concentrations, we're getting to below that 20 parts per million threshold that I mentioned the FDA would consider gluten-free. And keep in mind, we started with the amount of gluten that you would find in 2 slices of bread in this experiment. We also know that the enzyme works in beer. So we went out and bought a 6-pack of Chainbreaker White IPA, which is a wheat beer. And this beer has more gluten than its barley-made counterparts. If we add KUMAMAX to this beer, we see that this beer starts at a gluten concentration of about 760 parts per million. We see that the gluten level is well below that 20 parts per million threshold within 5 minutes when the beer is incubated at body temperature. And this is important because liquids empty through the stomach faster than solids. So you really need to be able to break down gluten in a liquid, particularly beer, since that's the liquid that gluten is most commonly found in, very rapidly. And we know it works at 4 degrees, which is fridge temperature. In this experiment, we went to Dick's, and we bought a hamburger and vanilla shake, and we brought it back to the lab. And this underwent the same treatment as the bread. And this experiment was meant to determine whether the enzyme can work well in the presence of large amounts of other proteins. So remember I mentioned that was one of our characteristics that we wanted in this enzyme. And so this experiment is testing to make sure that that's the case. So as a control, we used just a hamburger bun and water. And this control, the only protein in this meal is gluten. There's no other protein. And what we're looking at here, the black bars indicate how much gluten is present if you don't add any digestive enzymes. So this is really our starting point. The blue lines indicate how much gluten is present if you use these enzymes that are currently in clinical trials. And you can see these enzymes are not specific for gluten. And you can see that in the presence of just the bun, the gluten level goes way down. But when there's significant amounts of meat and dairy protein present, the ability of these enzymes to break down gluten takes a big hit. However, with KUMAMAX, if you zoom way into this 200 parts per million level, you can see that the enzyme is still maintaining really significant activity even in the presence of all of this other protein. So this is very encouraging to us. And finally, so we know that the enzyme breaks down gluten, but how do we know that the gluten that is broken down is no longer capable of stimulating the immune response? So this was a study that was done by our collaborators in Italy, in which they have isolated T cells from celiac patients. So T cells are some of your very important immune cells. And these cells are directly from the intestinal biopsies of celiac patients. So these T cells are highly reactive to gluten. So basically, when these T cells see gluten, they produce lots and lots of an inflammatory molecule called interferon gamma. And so what we've done is take gluten and treat it with KUMAMAX and see whether this KUMAMAX-treated gluten is still capable of eliciting this interferon gamma response. And what we find is that, in fact, the treatment of this gluten with KUMAMAX is able to eliminate this interferon gamma response. So in summary, celiac disease is a very debilitating illness. It affects almost 1% of the population. Non-celiac gluten sensitivity is a lot more of an ambiguous disorder. Much less is known about it. There are certain regions of gliadin that trigger celiac disease, and KUMAMAX is a potential treatment for this disease by breaking down these regions in the stomach before they get to the intestines and cause problems. And that's it.
Questions for Dr. Pultz? So it sounds like this is a really positive alternative to banning the Super Bowl, pizza and beer. Please, go ahead.
Hi there. I don't have celiac, but I have an autoimmune disorder, and my doctor had me go off gluten, and I've seen the antibodies, the self-attacking antibodies go down. Why is that? Is there an association with gluten and autoimmune response?
So celiac disease is actually considered an autoimmune disease. And this is because the disease triggers antibodies that recognize some of your own molecules. So do you know which antibodies it was that they saw decrease?
Thyroid.
Was it the anti-thyroid antibodies? Mm-hmm. That's interesting. No, I don't know the answer to that question.
Question over here.
Yeah, hey, that was a great discussion. Thank you. Hey, so my question is, yeah, looks like you've got some intriguing positive results with ground up Dick's hamburgers and that sort of stuff. But seriously, yeah, what's the next series of steps to go to more in-depth laboratory trials, animal trials, et cetera, all the way up to human trials? What's the next series of steps?
Mm-hmm. Yeah, so the next steps are to undergo the studies that are required by the FDA to go into humans, and then to go into humans. So what we're planning on doing is first a study in which we give the enzyme to people and look to see the amount of gluten that's being broken down in the human stomach. And we can do that by putting a nasogastric tube and place with aspiration ports in the stomach and the duodenum, the small intestine, to see how much of the gluten gets into the small intestine, because that's really the goal. And we can do that in healthy subjects, because as I mentioned, the amount of gluten that triggers the disease is 10 to 100 milligrams. So we'd want to make sure that we're breaking down the amount of ingested gluten to that level. So that doesn't even need to be done in celiac patients. That can be done in healthy individuals. And then the next step is, of course, to test it in celiac patients.
So do you go straight to human trials? Can you do this on bunny rabbits and animals and that sort of stuff?
So we're planning on doing this first in rodents. We'll need to test it in rodents. The FDA has very strict rules about when you can put something into people.
Question over here.
Yeah, and how long do you expect this to take, and when might you have something that those of us with celiac disease can actually use? And I don't intend that as a biting question. I have celiac disease. I'd love to have the stuff.
That's great. I get people contacting me all the time who have celiac disease. We're hoping that this will be available in about 5 or 6 years.
Can I get on your mailing list?
Absolutely.
What is the influence of the microbiota on the development of celiac disease?
So there's really no definitive studies that have addressed that. The microbiome it has, you know, its fingers in a lot of different pies. Certainly, celiac disease is a highly genetically linked disease, but it's not all genetics. So you have to have, you don't get the disease if you don't have the genes. But if you have the genes, that doesn't mean you're going to get the disease. You have to have the genes, you have to eat gluten, but there's some third component that's also involved, and nobody really knows what that is. And the microbiome is one of the hypotheses that's floating around, but nobody's identified the link yet.
Other questions for Dr. Pultz? So thank you very much. Excellent.